my name's Hayley and this is Susan, and welcome to Cancer Research Demystified.
So today's episode is all about blood. So when a cancer patient comes to the hospital, they might often be asked to take part in research, and often that would involve giving a few extra vials of blood. So usually what will happen is a nurse or maybe a scientist like one of us will come and take some extra blood samples from them and then sort of disappear off with them, and the patient never really hears more about it.
So what we want to do in this episode is talk about what happens when us scientists get that blood. So why are there so many tubes? Why have they got different colours? Why do we need that many anyway? How do we store it? What do we do with it? How do we process it so we can do some science on it? We're also going to touch on what the blood can tell us and why we're collecting blood in the first place.
Right, so we needed an example of some samples of blood to show you what we're going to do today, so we tossed a coin earlier and I actually won, so Hayley had to donate some blood for us today. I took it from her, but I was really gentle.
She was.
Yeah.
She was very gentle.
So we're going to use that now to take you through what we do with research blood samples. Okay, so we're in the lab now, and one thing that you'll hear us mention a lot is blood clots. So you've probably heard of a blood clot, but you may not realise how relevant it is to our research. I'm just going to go grab the blood.
So your blood will clot. When we stick the needle in to get it, you're introducing factors from your tissue into the blood, and your blood responds. It's super clever. It knows those things shouldn't be there. It forms a little clot to seal that hole, and the whole process stops, and the rest of your blood continues flowing. When we take the blood into tubes, if we just left it, because it's so dysregulated and it's not in your body anymore, the whole thing clots and we just get a sticky, clumpy mess in a tube. And sometimes that's not that useful. So these are Susan's blood samples, and as you can see they're all different colours, and there's a reason for that. So in a blood test you're normally looking for a biomolecule or something in the blood, and they can be stable in different conditions or they can be easier to find in different environments, and that's what these different tubes do, and we're going to go into what the different environments are. So first off, this is a purple one, and this is usually a whole blood tube, and when we get this back to the lab there's not much we do with it, we just want the whole of the blood. So what we're going to do is we're just going to mix it all up, we're going to divide it into smaller sort of volumes, and then we're going to put it in the freezer.
Okay, so then these other two, we're going to do a little bit more with them. So we're going to use a machine called a centrifuge to separate out different parts of the blood in these. One of them we're going to leave to clot, and one of them we won't. And that gives us two different things that you might have heard of, called plasma and serum.
Okay, so this is the one that clots, and there's some silica dust inside this that helps it clot, gives it a larger surface area. This one is the plasma tube, and this has a chemical in it all over the outside of the tube, and that stops the clotting process. That chemical is also in this tube, because as I said, if we're trying to get smaller volumes and the whole thing clots into a big massive mess, we're not going to be able to easily divide it up. Right, let's go.
Okay, so this is the tube that we wanted to clot, so we actually left this sitting around at room temperature for 30 minutes, and that meant all those molecules like stuck together and formed a big gross clot in it. So we're ready to spin this, so I'll pop that in the centrifuge, and then also the other one has been sitting on ice that's not going to clot. That also goes in here, nicely balanced and ready to spin.
Okay, so now our tubes are in here and they're spinning around really fast. So this machine is essentially like a merry-go-round. So imagine yourself in a merry-go-round. When you're spinning around like this, you can feel your weight being pulled to kind of the outside edge of the circle, and that's the G-force pulling you out. This thing will do the same. And what happens is it pulls the heavier bits of the blood. These heavier bits have more mass and are more affected by the G-force to the bottom of the tubes. So we have the heavy stuff at the bottom, leaving all the light stuff at the top.
Right, so it's a bit loud, as you can hear, and that's because it's really, really powerful. When we talk about G-force, we mean a lot of G-force. So, for example, a Formula 1 driver or some thrill seeker on like one of the world's fastest roller coasters, yeah, me, they're going to experience about maybe 5G, but these tubes in here are experiencing 2,000 G, so it's ridiculously fast. If you were in there, it would be separating out your blood too, it would break you.
And as you guys can tell, she loves roller coasters, so I actually think you'd do it.
I wouldn't want to. Yeah, no, I would try. Okay, so now that these are in here, they've got to sit there for 20 minutes spinning, so that gives us time to go for coffee. Okay, so we're back in the lab after our spin, and here are our two tubes, and they look a little bit different now.
Okay, so this was the tube where the blood didn't clot. It had a chemical on the outside of it, stop the blood clotting, and this one's the plasma tube. So you can see all the different layers. So all the heavy stuff's at the bottom. So the big cells, the red blood cells are here, that's why it's red. And then this is the gel. The gel was originally at the bottom, but it's moved up because it's specifically designed to be of an in-between density between here and here. And then at the top here, this is the liquid phase of your blood. So it's got everything dissolved in it that dissolves in, say, the watery bit. So fats, protein, sugars, even gases are in there. So this is a plasma tube, and this is what we use in our lab. Do you remember this tube that we use for the whole blood? And I said it had the same chemical on the outside. This can also be used to make plasma. And you get those two layers, but there's no gel in between, so it's not quite a clean separation. But what we do get is an extra, very thin white layer where the white blood cells are. That's your immune system. And that's used if you want to isolate those cells in particular.
Right, and then the serum tube does look quite similar to our plasma tube, so you've got these three distinct layers again. So down the bottom, instead of just having the red blood cells, we've got this big clot. It's pulled together all those cell types, all those proteins that help to form the clot. And the plasma on top has a little bit less in it because some of that's been pulled down into the clot. And again, we've got that gel in the middle. So although it's quite similar to plasma, the actual biology of what's going on in this serum layer is a little bit different, and we can do different types of experiments with it. And for both of them, what we're going to do now is just take this top layer, put it into little tubes, freeze it in the freezer, and then we can use it for experiments later. So once we've separated out all those little layers, we put them into tiny little tubes, put them away in boxes like this and store them in a freezer. And this isn't just any freezer, it's at minus 80 degrees Celsius.
So minus 80 is really, really cold. We need special gloves and special safety equipment. And the reason is, so if you think on Earth, the coldest temperature we've ever recorded is minus 89, so we're not far off with this. That was in like 83 in Antarctica, so it was pretty damn cold. So after we've collected and processed all that blood, we have a freezer full of it, and what are we going to do?
Right, there are so many different types of tests and research that we can use on great samples like that. But as an example, let's say Hayley is looking at a specific gene and she thinks that it might be involved in how cancer spreads throughout the body. So what she would do is look at the levels of that gene across all her different samples of blood and then correlate back to which patients did go on to have their cancer spread. And if there's higher levels of the gene from the samples that did spread, then she is probably onto something.
Another example could be Susan's looking at the diagnosis of cancer. Not only have we often got freezers full of blood from cancer patients, we also collect blood from patients that come in with all the symptoms, all the signs of cancer. They undergo a load of invasive tests and then they get given the all clear. And this is really useful because if we can find a difference in those blood samples, what we could do is help with the diagnosis earlier at the GP surgery, saving a few of those patients all those invasive tests, having to come into hospital, and all the mental angst that goes with that potential diagnosis.
Yeah, absolutely. And there are so many new cool tests like that you already are seeing hitting the news at the moment. Like every other week it seems there's a headline about some new magic blood test. And those wouldn't be possible if it weren't for these freezers full of donated blood that are all over the world in different research labs. It's really important that we do test these things rigorously, and that's how we do it. It's from collecting samples just like these. So with that, thank you very much for watching.
If you have any comments, we have a comment section down below. And thank you, and see you next time.
Bye!